I'm Kimberly McFarland, and today I'm going to show you about thin layer chromatography. So first thing I'm going to show you is how to pull capillary tubes. And first thing that's important is you want to make sure you're not wearing gloves because they are very flammable. And also make sure you're using double open-ended capillary tubes. So what you're going to do is put it in the flame and roll it back and forth, and then you're going to take it out and pull it. So just like that, and then you'll see it's nice and thin in the middle. Okay, so once you have your pulled capillary tubes, you're going to get a container and break them in half downwards into the container so that you don't send glass everywhere. And then you're going to get a paper towel and place it in the paper towel and then break it right where it starts to narrow. So you just push down gently and it'll snap off. Okay, so once you finish pulling your capillary tubes, you want to start setting up your TLC jar and plates. So with the jar, you want to make sure it's always covered so that your solvent doesn't evaporate. And you also want to make sure the level of the solvent does not exceed where you're going to spot your plates so that your sample doesn't run into the solvent. And also make sure there's an opening where your plate will go. You don't want the filter paper to be touching your plate. So now we're ready to draw on our plates. And you always want to handle it from the sides with gloves on. You don't want to touch it with your fingers. So you're going to draw dashes near the bottom, just on the sides, like that. And that's where you're going to spot your plates. So once you have your liquid sample ready and you have your pulled capillary tubes, you're going to spot your plate. And first I'm just going to show you on a practice plate what your spots should look like, and I'll give you an example of a bad spot too. So you want your spot to be fairly small, so you're just going to tap it real quick. That was a little too small, so I'm going to try again. There we go. And now I'm going to show you something that would be too big. Ooh, hello. So this is an example of something that is too large. Okay, now we're ready to check our spots by TLC. So we're going to place it in the jar, and you want to use the tweezers to put it in. Okay, and we're just going to let that run until it gets about 1.5 centimeters from the top, and then we'll remove it and check our spots. Okay, now that the solvent level is about 1 centimeter to 1.5 centimeters from the top, I'm going to remove my plate, and then it's important that you mark it immediately with a pencil as it will start to dry very quickly. And this is so that you can calculate your RF value later on. And now we're just going to let that dry, and then we'll check it out under the UV light again. Okay, now that we've read our TLC, we're ready to check our spots by UV light. So come take a closer look. So you want to place your plate under the UV light, and for this experiment it is normal to only have 2 spots. You may have 3 for your TLC lab, and that's fine. So I'm just going to lightly trace in pencil on the spots. I'm actually going to move this so I can get a better reach. And this is so that when you go back later, you know where your spots are, because watch when I take it out of the light, you can't see your spots anymore. Where did the spots go? Now that you have your spots clearly marked on your plate, you can calculate your RF value, which tells you how far each compound traveled. So for more information on this, you can go to page 263 in the lab manual, and it'll show you how to calculate your RF values. Thanks for watching. Good luck in your lab.